Hello friends, Ace Botany. Next episode. In this episode, we'll be discussing about principles of inheritance and variation, fifth one. So I will share the screen so it will be convenient. Then we can proceed. I think screen came. So shall we proceed? In fifth one of the principles of inheritance and variation, what all topics we'll be discussing? Mainly three topics: chromosomal theory of inheritance, then the famous organism for genetical experiment, Drosophila melanogaster, fruit fly. Then third idea, linkage and recombination, Morgan's experiments and result. And finally, as usual, 5 theory question, 5 neat model question with the key. So we can proceed. Starting with the chromosomal theory of inheritance. So before getting into it, we should analyze little bit background of it, some historical events. Mendel born in 1822. Mendel passed away in 1884. He worked in P, mainly in P, Pisum sativa, 1856 to 1863, so 7 years. Mainly 7 characters he took and 14 true breeding varieties. And Mendel's work was published in 1865 because it has to be published in journals. And it was published in Proceedings of Brunn Natural Science Society. Mendel passed away in 1884 after 16 years, that is 1900. Mendel's work was rediscovered by 3 famous scientists. You'll be remembering all this: Currens, De Vries, and Treschmark. And De Vries again published this work in Flora in 1901. And remember, Mendelian factor segregates in meiosis 1, specifically anaphase 1. It is a genetical question. It is a cell division type question. Now, chromosomal theory of inheritance, that is the first item we have to proceed. So what it deals? So we should analyze. Again, it will be history, and after that, the theory, essence of it. What is the aim of chromosomal theory of inheritance? Most basically, fundamentally, when we analyze, we can find this theory tried to establish connection between chromosome segregation and Mendelian inheritance connected with the meiosis. That is the essence, because we should know what it deals with. So connection between chromosome segregation, Mendelian inheritance in cell division called meiosis. By 1902, chromosome movement during meiosis had been worked out. And these statements given in NCERT, page number 65, 2024, March edition. And this famous theory, chromosomal theory of inheritance, was proposed by 2 scientists. Here you can find including photograph, Walter Sutton and Theodore Boveri, 1902, 1903, independently. And essence of it is behavior of chromosome were parallel to behavior of genes and used in chromosome movement to explain Mendel's law. That is why meiosis in connection with the Mendelian law. Sutton united the knowledge of chromosome segregation with the Mendelian principles and called it the chromosomal theory of inheritance. So Sutton originally combined all this. And chromosomal theory of inheritance, that is NCERT page number 67, March 2024 edition. Again, that statement is given. Sutton united the knowledge of chromosome segregation with the Mendelian principles and called it the chromosomal theory of inheritance. I think idea clear. What is the aim of chromosomal theory of inheritance? Who are the scientists and who combined it, which year, etc. Hoping that it is clear. Now we can proceed to second page. This is purely NCERT. And many people don't know what this diagram, why repetition like first and second. NCERT, when we read, we'll be slightly confused because too much details or the explanation they give on. Originally, what it means, it is connected with the meiosis, nothing more. So when we proceed to first diagram, that is this one, figure A, this one. When we analyze the diagram, what is the purpose of that experiment or diagram that I given here? That is chromosomal explanation of law of segregation. Very clear. And here you can find yellow, red, orange, green. And after space, then bivalence. Meiosis one anaphase, it will be separated. So same yellow, orange, yellow, orange, yellow, red, and orange, green. Here you can find, again, that will be separated. So once again, yellow, red, orange, green. Hoping that the color is same because red and orange slight difficult in NCERT. So this may be orange, this may be red. So yellow, red, orange, green. They will be segregating. And you can find when gametes are formed that yellow, red, and orange, green will be moving together. So remember, Mendelian factors segregate in meiosis 1 of anaphase 1. So when we observe the diagram, we may be confused, but nothing is there. From start, which combination that is available in gamete also. Nothing more. Simple diagram when we analyze in this manner. Now, second one when we analyze here. Aim I told here or written here. Chromosomal explanation of law of independent assortment. Any character can couple with any other character. So no combinations can be formed. Same thing, nothing more. So two possibilities. What are the two possibilities? Again here, orange, green, yellow, red. Orange, green, same manner comes down and 4 gametes. No mixing, nothing. That is possibility 1. Now possibility 2. Red and orange together, yellow and green together. So combination changed. In same manner, gamete came. So that indirectly explains chromosomal explanation of law of independent assortment. Any character can couple with any other character, that is all. This is the essence of it, but when we observe the diagram, we will be confused. So that is why the aim I wrote here in both experiments. Just a meiosis. First case, no mixing up. Second case, that is what is given in the parent same and it came. That means any character can couple with any other character. I think idea is clear. Any confusion comes, read these statements and it will be clear. So don't get confused, perplexed, nothing is there in that diagram. Then, here they given a comparison. That is another usual question. NCERT only, chromosome occurs in pairs, or both cases we can find occurs in pairs. Then statements are given. After that, they given a statement, a question. That is for our question. Can you tell which of these columns A or B represent chromosome and represent gene? How did you decide? See, chromosome means as such, chromosome separates. Gene means gene pairs, they will be separated. So automatically it indicates A will be chromosome. See here you can find independent pairs segregate independently of each other. And when we come to genes, there lies the answer. One pair segregate independent of another pair. So this is gene and this is chromosome. Popular question. Parallelism between chromosome and gene or a comparison between chromosome and gene. So last statement gives that is gene, this one, and this is chromosome. Here only one pair. I think idea clear. Now possibility 1, possibility 2, that is this one. And that is regarding this entire page. So when we externally look into NCERT, we may get confused, but this is the only thing available in that page. Hoping that it is clear to all of you. Okay, now we can proceed to next page. So many things when we observe externally, we may think it is confusing. Nothing is there. When we closely observe, it is simple. Okay, don't get perplexed. Okay, now comes experimental verification of chromosomal theory of inheritance by Thomas Hunt Morgan and et al. Et al. Means together. The famous organism is Drosophila melanogaster, fruit fly. You are familiar with it. And connected with this insect, many means at least one question we can anticipate in theory entrance, because this is one of the precious organisms in genetics, Drosophila melanogaster, because it has got many advantages that we studied in first episode of this chapter, organism selected for genetical experiment advantages or requirements. Now, details of this Drosophila. We are taking a usual Drosophila general item. Number of chromosomes, 2n equals 8. Total number of autosomes, it is 6, 3 pairs. So total number of sex chromosome, 2, that is 1 pair. And total chromosome in a gamete is haploid, that will be 4. 4 will be there. Autosomes will be usually 2, 3, 4. Sex chromosome will be 1. I think this part is clear. This is our previous question, that is why I wrote separately. Now regarding Drosophila, family Drosophilidae, order Diptera. Then Drosophila is often called Cinderella of genetics. Neurospora, it is widely used in genetics fungus. Drosophila of the plant kingdom. Guinea pig of the plant kingdom is another fungus. So same manner, Drosophila is called Cinderella of genetics because many ways we can utilize this Drosophila in experiments. And lifespan is normally 4 weeks. Fluctuations, variation, and different values are available. Normally 4 weeks, around 25 to 30 days. Then lifespan is completed in 2 weeks. Lifespan completion means from egg or the youngest stage to adult stage, different levels will be there. Like a pupa, larvae, and name. There will be different stages will be there. And that stages will be completed in 2 weeks. Lifespan will be normally in 4 weeks. That's the usual data. Exceptions may be there, fluctuations can be there. I think that idea is clear. And one more. This is male, this is female. You can clearly observe sexual dimorphism. And this is the chromosomal arrangement. Present NCERT, I think no diagram. Previous NCERTs, it is there. 4 pairs. First is the sex chromosome, first pair. This is male, this is female, X5, X6. Everything clear, I think. And sexual dimorphism also you can find there. This is male, this is female. Male is smaller. So automatically larger one will be female. And female has got ovipositor. It is connected with the egg. I think this complete area is clear. So shall we proceed to next area? Advantages of Drosophila. Theory question. Even how many correct, how many incorrect, that a type of question can be one need. Large number of offsprings it can produce. Organism is small in size, so easy to maintain. Sexual dimorphism is there. Life cycle is short, and variation among the offsprings. So these are the basic requirements for genetical experiments. All can be seen in this famous fruit fly, Drosophila melanogaster, that is usually attracted towards the ripened fruits like banana to consume yeast or fungus present in it. I think this page is clear, so we can proceed to next one. So gradually we are entering into Morgan's experiments with some basic terms: linkage, crossing over, recombination. So normally, what is the meaning of the term linkage? Linked or bonding. In simple terms, tendency of the genes to move together. And linked genes more seen on same chromosomes. Comparison, I'll tell you, if there are 10 individuals standing, and if they are tied together, they will tightly, individual cannot move alone. All 10, if they move together, they can move. Same manner. So tendency of the genes to move together. And usually linked genes seen on same chromosome. Such genes on same chromosome are called linked genes. And those present in different chromosomes are termed unlinked genes. Many terms are there in this area. But we have to proceed is mainly NCERT, even in Morgan's experiment. If genes are linked completely, complete linkage if it is there, only parent types or characters or offsprings will be produced. Types. If linkage is not complete, correspondingly recombinants also will be produced. I think that part is clear. Now we can continue. Term linkage was coined by T.H. Morgan, Thomas Hunt Morgan, and his main organism was fruit fly. Term recombination, or that event also given by T.H. Morgan. Morgan attributed more parental type. What is the reason? It is due to the physical association or linkage of the two genes on a chromosome. And term recombination to describe the generation of non-parental gene combination. This statement given in NCERT page number 67, March 24 edition. I think that idea is clear. What is linkage? What is recombination? Generally speaking, when linkage is more, recombination is less. When recombination is more, linkage will be less. So now, another famous statement, linkage groups in humans. It is 23. Linkage groups in male. It is 24. Linkage groups in female. It is again 23. So what is the criteria? Normally, linkage is haploid number of chromosomes. That is the principle. So human beings, 246, so haploid will be 23. That is why in humans, it is 23. Again, in female, linkage group will be 23. That is normal. But when we come to male, it is 24. Explanation I given here, why it is so. Because in male, two types of gametes will be there, X and Y. That will be separately counted. So 22 plus X plus Y. That is how it becomes 24. I think idea clear. Now comes Morgan's experiment. T.H. Morgan, Thomas Hunt Morgan, given a lot of contribution in genetics, specifically crossing over recombination, all this area. And even he received Nobel Prize in 1933. So getting Nobel Prize means, you know, the quantity of the work, quality of the work, depth of the work. But remember, NCERT, that level we are not proceeding. Now, two sets of experiment we have to study in this context. First experiment and second experiment almost ditto, only character is changed. When it gets changed, how the percentage, etc. will be changing, that I will show you with the NCERT diagram. So don't get perplexed again by looking into the diagram, that is so simple. Next page I will show you. So first experiment, body color and eye color. That itself is a question. In cross A, what are the characters taken? In cross 2 or B, what are the characters taken by the scientist Morgan? So cross A, experiment 1, body color and eye color. Body color and eye color, that is each case dominant recessive is, that is why each item I represented here. Body color is brown and yellow. Brown is dominant. Dominant will be represented by small letter with a superscript plus. That is a no. We studied in first episode. While eye color again, dominant red, that is red, recessive white. Red is represented by small w plus, superscript plus. White is small w. For time being, we can observe that one first, then we can come to this one. This is a famous experiment. When people observe this, they will be confused. I told you, we are not going to the depth of original experiment of Morgan. That is very deep, Nobel Prize winning work. So you know the quantity, quality, everything. So we can proceed. Simple manner will explain, so easy to remember. Cross 1, body, everything written there, you can find. Body color and eye color. Body color, that is brown, yellow, eye color, red, white. Plus sign indicate it is dominant. So in this case, you can find female is shown there, it is male, female is recessive. So recessive character means yellow and white. That is why no superscript you can find. So automatically male will be dominant or wild type. Y chromosome, nothing to do. Most simplest manner if I explain this, even though it is not perfectly right still. From here only one type of gamete we can get, that is small y, small w. Here two type of gamete, Y plus W plus and Y chromosome. So one of these couples with the Y plus W plus and Y chromosome, that is what written here. That way when you proceed, it will be easy to remember, easy to do everything we can do. If we correctly explain it, it will be little bit difficult. So in F1 generation, what we can find, female become now dominant. First parent we can find male was dominant, the character came to female, F1, and male is recessive. And now from here, two types of gamete, from here, two types of gamete. Here YW, Y plus W plus, here YW and Y chromosome. And that 4 will be just coupled here. That is why I told it is so simple. If you proceed this method, that is pure parent type you will be getting. Parent type in the sense, any character if you check, you can find either here or here. No variation from parental type for this 4. That is why they return parental type. Now, when we come to here they given recombination type. So no parental type you can find. And how this recombination comes, it is so easy. See, here you are getting Y plus W. How it is possible when we cross these two? Y plus W and Y chromosome. So these two will be over. Second one, YW plus, same manner female side. If you remember this method, it will be easy to remember, easy to draw any question you can do. If you proceed in a scientific manner, that is the right one, but it will take time. It will be slightly confusing. That is why this method I explained. I think this part is clear. Now, when we come to parental types, it is 98.7%. Recombinant type, it is just 1.3%. That means here, recombination is less, parental is more. Further explanation I will tell you after finishing this one. Now, second one, crossbreed. What are the characters I written here? Eye color and wing pattern. Wing pattern is normal. Normal means wild type or dominant, M+, and miniature means recessive, small. Same type of cross. Here also you can find female is recessive, just like this one. Male is dominant. When we come to F1, female dominant, male recessive, and these two will be crossed. You'll be getting 4 parental type, 4 recombinant type. Just a ditto like this. So once you get this idea, this will be easy. And if you got both ideas, no need to by heart even. If you get the parent, you can write up to this one. Now this is pure parental type. No recombination, nothing. Here you can find mixing is there. And in that case, you can find parental type is comparatively less comparing with this one. Here it is only 62.8. And here it is 37.2. First cross you will be getting parental 98.7 and recombinant 1.3. I think that two idea you got. Now we can come to this part. Cross A. Everything I written there. Parental type 98.7, first cross. Recombinant type 1.3%. Why this parental type is more on cross A? Because that two genes, they are tightly linked. So recombination is very less. If completely linked, no recombination. It is tightly linked. That is why slightly recombination we got. And how it is taking place, because that I will tell you last. Now this side. When we come to second cross, parental type is 62.8, recombinant type is 37.2. And these two genes are linked loosely. That is why recombination is more. And how or why it takes place? Because you can find genes here. They are close. Genes here, far away. Far away means crossing over is possible, more recombination is possible. So the distance actually matters. And this idea that is utilized by student of Morgan, namely Alfred Sturtevant, by recombination frequency between genes located on same chromosome. This idea is utilized to map their position in chromosome. So Sturtevant prepared first chromosome map of fruit fly using the recombination frequency. How far it is placed. For that, there is a simple formula. Total number of recombinants by total number of progeny into 100. Remember, these all, we got the data, then we are only proceeding. For them, they have to do all the experiments. That's why I told you why Morgan got Nobel Prize in 1933. It's a Herculean work. And this way we can calculate recombinants. You will be remembering how we calculated dihybrid cross recombination frequency. 6 by 16 into 100. You'll be remembering same manner. And there is a unit for it. In eukaryote, unit is centimorgan. And in prokaryote, it is map unit. So eukaryote, it is centimorgan. Prokaryote, it is map unit. So as such, when we look onto this diagram, we may get perplexed, but nothing is there. And remember, this figure belongs to NCERT figure 411. So usually questions will be based on NCERT. Too much outside question, normally they are not asking now. They may proceed into depth, but outside chance is very, very less. Now after this, Usually 5 theory question, 5 NEET model question with the key. I think it is clear. So what all things we learned in this episode, just we can rewind and proceed. Starting with this, what all topics we'll be discussing, some historical events, then Mendelian factors segregate in meiosis 1, then Sutton and Bovary, chromosomal theory of inheritance, what is the aim, what is the idea, then 2 sets of NCERT diagram, where this one explains chromosomal explanation to law of segregation. Then after that, the chromosome gene comparison. Then diagrams, their explanation NCERT. And the second diagram, that is this one, chromosomal explanation of law of independent assortment. After that, we entered into third page, Drosophila, complete details, family, then lifespan, life cycle, difference between these two, then syndrome of genetics, total chromosome, total number of autosome, total number of sex chromosome in a gamete, and autosome 2, 3, 4, sex chromosome 1, and their position, then how female differ from male, then advantage of Drosophila. After that, linkage, then crossing over, recombination, meaning of the term linkage, how it is broken through crossing over and produce recombination, new combination, and who coined the term linkage, T.H. Morgan, famous organism is fruit fly. Then after that, linkage groups in humans, males, females, why it is 24 in male. Then experiment of Morgan, experiment 1, 2, or A, B, character, body color, eye color, wing pattern, and the idea behind the experiment. When we observe this, will be confused, but nothing is there, I told you. Then all characters given. After that, cross A, cross B, why recombination is less in cross A, why it is more in B. And after that, Strutman, by using these principles given by Morgan, how he developed the chromosome map. Then formula to find recombination frequency. Then after that, it's a unit, that is Morgan in eukaryote, map unit in prokaryote. Then as usual, 5 theory question, 5 NEET model question. If you like this episode, subscribe all, like it, share it, comment on it. We can meet later. Thank you. We can wind up. Thanks.